but I can also do stuff if I have to. Help session on Thursday evening at 8:30 p.m. on Google Meet. Office hours by appointment today, so let me know if you want to come see me today about anything, lab report, exam studying, whatnot. Just let me know right after class. And then your lab reports are due on Saturday at 11:59. Questions on stuff we've got going? Okay. Practice exams are up. There are extra practice worksheets up. Okay. You might want to look at, since we didn't do a quiz on putting the pieces of membrane lipids together, you might want to look at the example worksheet for that, which is an old quiz. Okay, often people take that quiz and they're surprised by it or they're not really ready for it and they do poorly on the quiz and then they get it together for the exam. But you didn't have the chance to get scared by the quiz. So do look at that. There weren't any questions yet. Chloe?
Can we go over actually like a few of those examples?
How we would do that?
Yeah.
Sure. Let's say, so let's grab an old exam for that. I probably have the old exams. Does anybody know off the top of your head, do both old exams have that?
Yeah.
I know at least one does. They both do?
Yeah.
So it doesn't matter which one I pick. Okay, so we want exam 3 something. We'll take this one. Oh, well, we'll take a picture of it, I guess. All right, there we go. All right, so I just want the molecules. Go ahead and grab the pieces and drop them over in the notes. And make them big. Okay, so you'd be given the way that would work is you'd be given, the question is usually draw a blah and write a sentence on what its function is. Okay, so it could be, and what I want you to remember is that really in the name it tells you what pieces go into it. So you need to be able to recognize what the pieces are here. So this is sphingosine. You need to know where to hook them up, right? So our bases are going to be, we're either going to have a base of a sphingosine or a glycerol, right? And then our polar head group is going to be, if we're doing a membrane lipid, is either going to contain a phosphate or it's going to contain a monosaccharide of some sort. Okay. Which one is that? Which monosaccharide?
Oh,
You'll want to have that glucose, yes. Okay, so that's glucose. So let's say you're asked to draw a glyco. Okay, so that's the polar group, a glycosphingolipid. All right, it's right there in the name. Glyco, it means we've got a monosaccharide that's going to be on there, and then we've got sphingo, which means we've got a sphingosine right here. So a glycosphingolipid is going to start with the sphingosine, which, like you, I don't have memorized either for the structure. But so I'm going to go ahead and try to get this right. 2, 3, 2, 4, 6, 8, 10, 12. One more. 2, 4, 6, 8, 10, 12, that. And then if I look at this, I then have a hydroxyl, and then it's going to go up. And I can actually, I don't have to do this in a, all of them pointing the same way. Like the tails, this is one of our fatty acid tails, just comes straight from the sphingosine, but I don't need to draw it so that they're going in the way they might sit in the membrane. So I'm just going to draw it like it's drawn here because that's going to be easier for me. And then I need to figure out where I hook up things. So I've got 2 places to hook up things here. What goes on the amine and what goes on the hydroxyl?
the amine goes with the fatty acid.
Yep, the amine goes with the fatty acid tail. So I can draw it now out of fatty acid. I have 2 fatty acids. I can choose whichever one I want. 2, 4, 6, 8, 10, 12, 14, 16, 18, was it? If you just get close, there's no way I'm counting on the test. But 2, 4, 6, 8, 10, 12, 14, 16, 18, okay? And then up here on the hydroxyl, that's where I'm going to put my polar group. My polar group is supposed to be glucose, so I'm just going to go ahead and hook a glucose up to it. And it doesn't matter, I mean, I'm sure it does, but for our purposes, I don't care where you hook it up. Okay, if this was an oligosaccharide, if this was the starting bit of an oligosaccharide, as it often is, right? We're just doing a simple one here, but oligosaccharides can be connected by any of those hydroxyls, even though this one has a special reactivity. Any of those hydroxyls, and you get all sorts of weird branching structures. Because remember, our oligosaccharides, if I was to build a bunch of other sugars on there, are involved in information. They're cells talking to other cells. You're knowing self from non-self, things like that. Questions? You see what I did? What that was? Okay. And so glycosphingolipids, what are they involved in?
Signaling.
Yes, involved in signaling. Very good. Okay. Questions on how you do this? So we could have let's say we have a glycerophospholipid. Go ahead, Bridget.
Oh,
Do you want a different one?
no, does it matter is it always going to be the one carbon that it attaches to on the?
It doesn't matter.
Oh, it doesn't.
I'm telling you, it doesn't matter for the purpose of my grading this. In nature, it does matter. I wouldn't be surprised if it was almost always the one carbon, but oligosaccharides, when we think about oligosaccharides, they are not always attached by the one carbon. They're attached by all sorts of carbons to give you these really interesting structures. Okay, so let's go ahead and we'll try a glycerophospholipid. Okay, glycerophospholipid, it's right there in the name again. I did switch which side I did, right? Before it was the glycosphingolipid, this was the polar group, and that was the backbone, and now that's the backbone, and that's the polar group. You should be able to pick those words out of there. Kind of these things get swapped around depending on whose book you're reading. But the words are right there. So glycerophospholipid, we're going to start with a glycerol. Okay, so we got this one right here. If there's room on the test, probably, you know, if I will ask this question. Where I ask this question, I probably won't leave you a lot of vertical room versus horizontal room, but if you have it, great. Turn your paper sideways. But I'm going to take my backbone here, and I'm not going to bother to do anything special with it. I'm not going to rotate it or anything. Okay? And then I need to connect things to each of those hydroxyls. So I'm going to go ahead and I'll just erase, since I'm going to not have those hydrogens in the end anyway. A glycerophospholipid is going to have a phosphate for a polar group, right? Which hydroxyl does it go on? On one of the outside ones, not the middle one. Okay, just not the middle one. So it doesn't matter which one of the other two you choose because there's not a difference. And then I'm going to go ahead and I'll put a phosphate here. You will have to draw all atoms for this, okay? And these things are often more complicated than that. There's a phosphate, maybe there's an amino acid attached to it as well. There are more greater things you can build onto it, but I would just do the phosphate. I would just take the simple one and move on. And then I need to attach 2 lipids for the tails, okay, the fatty acid tails. And so I'm just going to use, what was it? It was 18 carbons, 2, 4, 6, 8, 10, 12, 14, 16, 18. 2, 4, 6, 8, 10, 12, 14, 16, 18, okay? And they can be the same, they cannot be different, can be different. It depends on what you're looking at in nature. We used to have a nutmeg lab, and what happened with the nutmeg actually has, the nutmeg nuts have a fat, it's a triacylglycerol in it, but the triacylglycerol has, it's all 14 carbon fatty acids, which is kind of cool. Questions? All right, so that's the sort of stuff I'll expect for that. There is a practice worksheet. You should go ahead and try it. Oh, what are glycerol phospholipids? What's their role? They're structural. Structural. There we go. They're structural. So essentially, they're making up the bulk. In animal cells, they're making up the bulk of the membrane. Right? There are going to be other special lipids in that membrane as well, but they're going to make up the bulk of it generally. All right. Other questions then? Yes.
Can you go over the sodium potassium membrane?
Sure. The sodium potassium ATP transporter? Yes. Or ATPase? I want a picture of it. Maybe it's here. There we go. All right, so this thing is awesome. Whoops. Not made better by fatty acid parts, though. And so you should be able to describe the cycle for this one in detail. It's an important transporter. Let's go ahead and make it small enough that it's all on the screen at once. I think I'm going to leave it there. I don't need you to read the words right now. When you're studying, you can read the words. Okay, let's walk our way through it. So we got this enzyme. It's an enzyme transporter, right? Okay, but it's you got this transporter, and it has two conformations. It can be open to the inside of the cell, or it can be open to the outside of the cell. Now, the role of this thing, what it does is, the first thing you need to know, right, the most basic thing, which doesn't involve the mechanism here, is that what this is going to do is, if we have this cell membrane and we have a sodium potassium ATPase here, that what it does is it's going to pump for every ATP used. So let's use an ATP here. Let's reflect. I'm not going to test you on this, but let's accurately reflect the fact that the ATP, all parts of the ATP stay in the cell. Okay, so it's going to use an ATP and a phosphate, and for every one it uses, it's going to pump 3 sodiums out and 2 potassiums in. So this has specific implications. All right, your body uses this to great effect. Like I've said a number of times, this is using about 25% of your resting energy. All right. So first off, let me ask some questions about this. It pumps out 3 sodiums and 2 potassiums. So what does that do to the charge inside the cell compared to outside the cell? It's going to be more negative inside the cell, right? Because we pushed positives out. So we push net positives out. Cells, in general, are negative on the inside and it's positive on the outside. There's a potential there. And that potential is good for things. That potential can power secondary active transport, right? Remember, secondary active transport is where we let something in and we bring something else in with it. So something's moving down an electrochemical gradient to help push something else up an electrochemical gradient. Does that make sense? Okay. But that's an important part of this system. That's what it gets used for. Let's talk about how it works. So right now here at the top, it's open to the inside of the cell. When it's open to the inside of the cell, it has a high affinity for sodium. Okay, so that's here, and that's also down here. So it's open to the inside of the cell. It's a high affinity for sodium. Sodium binds to it. It then opens the outside of the cell, and now it has a low affinity for sodium, but it's only open to the outside, so the sodium pops out. Okay, so we're picking up this sodium. We open to the outside, pick up this sodium. We open up to the outside, and that sodium pops out because we now have a low affinity when we're in this conformation. So we could add that over here. We could say low affinity for sodium. And that's right here. Yes?
You said this shows secondary active transport because I wrote in my notes
This is
primary.
primary active transport. It is used to power, the results of it are used to power secondary active transport.
Like what?
Put a pin in that. We'll
Okay.
come back to it. Okay. So we're open to the outside. Okay. So now we release those sodiums. At the same time, the reverse is happening. So when we have low affinity for sodium, we have a high affinity for potassium. All right, so we have a high affinity for potassium here. And so what's going on here is now we're open to the outside. And so potassium binds when you're open to the outside because that's when you have a high affinity for potassium. And then you flip back to the other conformation. The ATP is helping to power, or is powering it opening and closing. So that's why it's primary active transport. And when it opens the inside, then we have a low affinity for potassium and it releases potassium on the inside. Oops. Just along with the other arrows. Okay. Yes?
Is this like what is it, hyperpolarized or something? Or just like
It's going to polarize the membrane. It's not hyperpolarizing. It's just polarizing our membrane so that that can be used. Yes?
It's using the ATP in order to do this?
Yes. And so in terms of powering it with ATP, this ATP comes in and it sticks a phosphate on, which changes, phosphate has two negative charges on it, it changes the shape of the enzyme. And then the enzyme switches conformation, it holds open, and there's going to be some sort of phosphatase activity here on the enzyme. And so it's open, and then the phosphatase eventually gets to this phosphate, and then it comes off, right? So you might have a slow phosphatase activity. So it stays open for a bit, then it closes. Or opens the inside, really. Questions? Okay, let's go back to one of those exams is going to have a secondary active transport question on it, I think. Is it this one? I'm looking for a pretty picture of a membrane. I'm not seeing it. So maybe let's look at the other exam real quick.
On the last page.
Is it? Thank you. Okay, here we go. We've got a transporter. All right, so we've got a cartoon here and something's being pumped in. This may not be useful, actually. This looks like primary active transport to me. So the protein transport carrier channel, it's going to be a carrier because it's binding. It's not just a hole through the membrane, right? If it was a channel, we just have a hole through where we wouldn't be binding to something and changing conformation. The transport's an example of primary active transport here because we're using ATP, just like we did with the sodium potassium ATPase. And then given your answer to B, the substance being transported is most likely to be doing what? Go ahead and look at these and think what your answer. Consider natural selection in your answer. Consider efficiency. How many people think A? How many people think B? How many people think, well, C or 3, whatever, but yes. Okay, so it's going to be moving it up an electrochemical gradient because you're using ATP. So you wouldn't just use ATP to use ATP, right? Your cell is not going to burn ATP unless this was something designed to generate heat, which nothing like things that generate heat aren't moving things necessarily. It's going to be moving it up an electrochemical gradient, uphill because it has to use energy to do it. Yes?
Can you go over secondary active transport and what a symport and import are?
Yes, yes. So let's go back and look at, there's a nice figure for this. It's not that important. So let's just first talk about what symport, antiport, and the other one, which was that? Oh yeah, uniport. I was trying to think of what the two in the same direction. It's symport. Okay, so we have uniport is one thing. We'll just make it A moving through. Okay, symport is going to be two things moving through and then moving in the same direction. This could be A, and this could be B. And then we have antiport, which could be A. A is going to be moving in one direction, and B is going to be moving in the other. Whoops. Let's make B green. So the sodium, potassium, so this is independent of energy usage. Okay? So the sodium, potassium, ATPase is which of these? It's antiport, right? Because we've got one moving one direction and one moving the other direction. And any of these could be, well, any of these could be anything, right? Any of these could be passive, any of these could be active, secondary active, right? Any of these could be active. Yes.
So it doesn't matter if it's a channel or a carrier, they can still.
Yeah, I mean, you're not probably going to get channels doing this.
Okay.
That makes sense. So, what was that?
Would it be like just uniport because channels are typically specific?
Channels are not very specific,
Sorry.
right? But it would be it'd probably be uniport because you have a channel lets in water. It could let in more than one thing, but probably not at the same time, right? It's just stuff going through a hole for a channel. For a carrier, these are probably carriers because you're going to specifically let in one thing and another thing at the same time. But it could be passive. It could be going down and up an electrochemical gradient that wasn't generated by something else on purpose, right? So an example of passive transport where you have an electrochemical gradient is your gluten-1 transporter for your liver cells. Your liver cells, if there's more glucose outside the liver cell, it comes in. If there's more glucose, if there's more glucose outside the liver cell, it comes in. If there's more glucose inside the liver cell, it goes out, just naturally.
You had mentioned like the secondary active transport. Is there a specific type of transportation for that?
Well, so secondary active transport is going to be either symport or antiport. It's going to be one of those two. Let's see what the internet has for us. Unless we have one of those in one of the old exams, but I think we don't have. Do we have a? I feel like I maybe saw that. No, we didn't have that on both exams. So, otherwise, we'll just go to the worksheet. I think that the worksheet I gave you had possibly an example on it as well. Let's do secondary active transport. And let's go ahead and look at the images. And just looking for one I think is good. Oh, here's a good one. Oh, it's a YouTube. All right, can I make this big? I don't want to show you the YouTube video because I don't even know what it's about. Well, I mean, I know what it's sort of about, but nah, it's okay. I'm going to point to it real quick, and then I'm going to try and find a different one. All right. So I could, but you know, it's going to be an ad, and then it's going to be it's. I'm not hopeful that that will be a super efficient use of our time. Let me do this. Okay, so what we've got here is we have this, whatever this glucose transporter is. This is something, they don't name it, so, but what they've got is you're trying to pump a lot of glucose into the cells, okay? And now cells naturally have a lot of sodium outside. Why is that? Sodium, potassium, ATPase, which is actually even shown right here, okay? So we've got sodium, potassium, ATPase here, pumping sodium out. You're trying to concentrate the glucose in your cells, so maybe this is in your intestine, if we were thinking about that. And so you could help bring that glucose in up a electrochemical gradient. In this case, it's just a chemical gradient, but you're using an electrochemical gradient to bring it up, right? It's just a chemical gradient for glucose. Why?
Does it have a charge?
There's no charge, right? Okay. But we're using an electrochemical gradient because sodium is charged, right? There's more sodiums outside and they're charged. So it's negative in here, it's positive out here. The sodium wants to come in, and this thing is set up to let the sodium in and bring the glucose in at the same time. Okay, and so the sodium is moving down an electrochemical gradient, so energy is released, and more energy is released letting it in than is consumed in bringing the glucose in up the gradient. Now, the reason why you want this is if this is in your digestive tract, you don't want to like let a whole bunch of glucose or whatever useful molecule that you're eating, right? You don't want to let a whole bunch of that just pass through you because it's not moving up a gradient to get into your cells. You want to use energy to make sure you get as much of it in as possible. Okay. And from the perspective of energy here, if we want to even be more specific about it, if we use an ATP, if we're only letting one sodium in, okay, we're only letting one sodium in to do this, if this is really what's happening, that's a third of an ATP, right? Do you agree? Okay, it's a third of an ATP to let that sodium in because it took one ATP to push 3 out. Okay, so it's a third of an ATP to bring that in, and one glucose molecule, if you're getting enough oxygen, is worth 32 ATP. This is well worth your effort if you're a cell to do that. Does that help with what secondary active transport is?
So you use ATP to make a gradient, and then whatever.
Yes, yes.
With it?
What was that?
It's like Symport, or can it also?
It could be antiport. I don't know that I can give you a real life example of it, but there's no reason this couldn't be that we couldn't have potassium leaving and glucose coming in at the same time, right? Both of them are used to generate, are used in that gradient. We get a gradient for both of them in the cell. But that gradient was generated, okay, it's not evolution doesn't think, but the reason why you're spending 25% of your energy pushing sodium out and potassium in is so that you can use that to do work, like bring things into the cell. Does that make sense? What that is? That's an example. Is that enough?
So wait, in the ETC, is that secondary? Because you're creating a gradient of proton.
I'm not going to call that secondary active transport. You're using the gradient to do work, but not to bring things in.
So are you bringing through?
You're bringing through a proton and you're making ATP.
Okay.
Now I don't know that I can find the figure very easily, you actually let you need to bring ADP back into the cell, and I think you use a phosphate to bring it in. You actually use a proton to bring it in, bring either the phosphate in or the ADP in, right? Because you make ATP in the so let's pretend it's phosphate, but I don't remember off the top of my head. I'll know it second semester, whichever one it is. It's either the phosphate or it's the ADP. But you've got your mitochondria, and what it's doing is it's making ATP, right? And then it shoves that, if this is the mitochondrial membrane, it sends that out of the cell, and out of the cell, that ATP gets made to ADP plus phosphate, right? It gets used, it becomes that, and now we have to bring it back in. And these get brought back in. Or actually, maybe, I think the ATP maybe go at the same time. Sorry, I'm going to have to look at my notes to remember exactly what this is. But there is a proton in bringing, part of this process involves a proton bringing it back in. And it may be that we have an antiporter that actually has the H plus involved as well. Sorry, I could find the figure, but it would take me 4 minutes or so. Where is that in second term? Sort of near the end. Yes?
I have another question about something different. So when we were looking at the graph, I was confused on like when the x-axis is the concentration of the substrate and then the y-axis is the initial.
Right. So we're talking about kinetics now.
Yeah.
Yeah. Great. So good question.
I was also confused like the difference between graphing the initial and the max because I just didn't understand.
Okay. So I'm not sure. I'm going to start talking, and I'm not sure what to address exactly. So I'm going to start talking, and then you can help guide me maybe. So this is a Michaelis-Menten plot, right? And this comes from our Michaelis-Menten equation that looks like this. Okay, that's our Michaelis-Menten equation, and we had our 45-minute derivation we did on it. You won't have to do the derivation. You would maybe have to do graphy things, okay, to demonstrate an understanding of that. But so a plot for this looks like this, okay? I know that because I've seen it a lot of times, but I also know it from my equation. Equations are your friends here, okay? I know that as I increase my substrate concentration, that it dominates the Km, right? When substrate concentration is really small, let's say, the denominator here stays the same, right? If it's small relative to Km, this is unchanging. If this doubles, then that's going to double. If the amount of this I add to that is very small. But then as I get to very high substrate concentrations, where substrate is much higher than Km, when that doubles, and this is very small compared to those two, then nothing happens, right? And so that's why this thing starts out, I draw them like this, but it's linear. It's as you double concentration, double, and then you start getting to the point where you're near the K, getting close to the Km, and then it's going to curve over. And so this is, in your experiments, what you did is you varied the substrate concentration, and then you measured something that was proportional to the initial velocity, right? So the last ones you did, you measured a fluorescence, which you were measuring as a current, right? No, it was potential. The potential, so in millivolts, right, on your voltmeter, okay? So you measured that as a potential on your voltmeter, but that was proportional to how much product was being made. So now let's talk about where Vmax is, right? So Vmax is the maximum velocity you can get for that amount of enzyme. Remember, you didn't vary the amount of enzyme in your experiments. You varied it to figure out what a good amount of enzyme was, and then you set it and held it constant, and you only varied your substrate concentration. And so for a given amount of enzyme, you're going to have a Vmax. So I'm drawing this, let me do it just a little bit. The Vmax is not, I don't want you to draw your Vmax necessarily at the top points. I mean, if it's clearly leveled off all the way, but let's say Vmax is here. So what I'm saying, looking at the graph I drew, even though I kind of drew it, it's level up, I think it would get up to here eventually. So there's my Vmax right there, okay? And that happens because, let's see, that happens. I mean, why we get a Vmax is we've got the equation that tells us we get a Vmax. We also know from our model, you've got enzyme plus substrate is going to go to make an enzyme substrate complex, and then that's going to make a product and release both. Well, it's going to release the product. So I only have so much enzyme here. And an assumption we make in these experiments is that I have a lot less enzyme than substrate. Okay? The enzyme is a catalyst. There's no point in having an enzyme or calling it an enzyme if you have as much of it as substrate, really. It might still turn things over, but you just don't have, there are very few situations where you would ever have as much enzyme as you have substrate. And I can't think of any off the top of my head. We've got enzyme plus substrate, but as I increase the amount of substrate I've got here, we can use Le principle to think about this. We're going to shove more of that enzyme into the enzyme substrate complex, and eventually I get to the point where my enzyme is just all bound. It's all bound by substrate, okay? And so that's why I get a Vmax. I have a maximum velocity. I can't go any faster. Yeah.
Yeah. So what if you were going to graph like instead of V initially you do Vmax on the y-axis? Like how
Sure.
would the graph change?
That's a great question. So I've got my substrate concentration here, right, versus Vmax. And these graphs always assume that we have, unless you've got enzyme on one of these, you're going to assume that you're varying substrate, you are not varying enzyme, right? Nothing is being, you're going to assume that whenever I do a graph like this, nothing is varying except whatever you've got on your x-axis, right? So what is Vmax for a given amount of enzyme? How does it change with time? It doesn't, right? So Vmax is a constant, and so it would look like that.
Because it's the same amount of enzyme.
It's the same amount of enzymes, so the maximum velocity you can achieve does not change.
But isn't it going to be smaller if there's less substrate?
Isn't the Vmax going to, it is not, your assumption here is there's always more substrate than enzyme. So Vmax does not affect, the fastest it can go is not affected by how much substrate you have. It's just a constant. It may not be going that fast. With less substrate, it will not be going that fast. Other questions while we're doing this? This is sort of graphy thing stuff. What different question?
Can we look at go through the other forms, but substrate and time and
So give me one. So you want to do substrate?
Yeah, substrate and time, and then the other one was the concentration of enzyme plus concentration of enzyme substrate complex.
So let's do one at a time here. So substrate versus time. So we've got a reaction going on here. We start out with a lot of substrate, a little substrate. Okay. We start with a lot of substrate, and do we end up with a lot of substrate or a little substrate? Okay. When I have more substrate, does my reaction go faster or slower? Or does it not matter? It goes faster, right? The more substrate. So remember, our rates, our V naught is going to be equal to our K cat times our enzyme substrate complex concentration. And if we look at our reaction up here, the more substrate I have, the more enzyme substrate complex I'll have, right? In terms of Le Chatelier's principle.
Mm-hmm.
So it goes faster at the beginning. So what we're going to end up with is something that's going to get really steep and then it gets less steep. Because as the substrate goes away, so substrate's going away really fast, and as the substrate goes away, it goes away slower because you're making you have less enzyme substrate complex. Okay, what was the other one, Bridget?
Total enzyme concentration plus enzyme substrate complex concentration and time.
Okay, so it's enzyme concentration.
E plus ES.
E plus ES versus time. Sorry. That's just, this one's just kind of mean. What is E? What is E plus?
It's total enzyme.
It is.
They just cancel each other.
No, they don't cancel each other out.
So it stays the same, doesn't it?
So it stays the same. It's the total enzyme in solution, right?
Okay, so I was just overthinking it.
I mean, it may be one of the other tests. I generally kind of give a couple easy ones, then maybe a middle one, and then a kind of really complex one. So I don't promise not to do it, and I very well may have done it in the past.
Oh, that's why.
Is that one of the tests?
Yeah.
Yeah. Okay. Well.
I was just overcomplicating it.
Yep. So it's the same as that, and it's the amount of enzyme you threw in, and none of that enzyme goes away. Okay.
Can we do like the transition state where it like talks about which one is the inhibitor based off of like your compound?
Right. Okay. So let's talk about theory, and then let's go to the test question. You're looking at an old test question there, probably, because that will have it built up in terms. It'll have some structures. So when we think about a reaction, so we've got a reaction coordinate down here. We've got energy over here. Right? We're going to have our substrate. We're going to have our products. This will be our uncatalyzed reaction. And this will be our catalyzed reaction. Okay. The stick-ace figures are they're a little more complex. They're a little better. We're not going to really worry about that so much. But if we think about this, so how does the enzyme, there are multiple things going on, but what's one of the ways the enzyme lowers this transition state energy?
Lower activation.
It lowers the activation energy, right? And what it's doing is it's binding to the enzyme binds tightly to the transition state, whatever that transition state is. So it binds, it makes bonds to that transition state. We could say it stabilizes it, but it's making bonds. When you make bonds, you release energy. So you make bonds to that transition state and you release energy. Okay, and so that's why this is lower than that. If we bound tightly to the substrate, we'd pull the substrate energy down. We'd pull the complex of the substrate energy down, and then it would look like this. I'm going to erase this after I draw it. But if we bound tightly to the substrate, we'd pull this down here with our binding, right? And then we'd still have to get all the way up here again to come back. So if you bound tightly to the substrate, you'd make a little complex here with the enzyme and the substrate. You'd stabilize the substrate. You make this hill higher to get over. Does that make sense? Okay, so enzyme has to bind very tightly to the transition state for this to work well. And so then let's go look at that test. Which test is it?
The last one. The second.
So
first one.
This one?
Yeah.
Got it. So we need to bind tightly to that transition state. I think it's up here. Here it is. All right. So for this problem, I've got the reaction right here that's going on. So this is, what are we doing? We're making inosine? We are making inosine. Okay. So we got adenine is going to react here. And it looks like we have a nucleophilic attack. We have bonds flying around and all over. But this is what my transition state looks like, right? Do you see? I probably need to blow that up a little bit. Okay. Do you see in our transition state here? I don't need to worry too much about what's going on exactly with the mechanism, but that transition state it's not aromatic here, right? Okay. And we've got a sp3 hybridizing. hybridization here in transition state. So that stands out to me, right? We start out with something that is sp2 hybridized, we go sp3, we go back to sp2. All right, that's a big deal. Let's look at our possible inhibitors and see if we can just pick off one that is sp3 there and be done, or whether we have to find some more things that look similar. All right, so here are our analogs. Which of those looks most like it? See, we have this one's sp2 hybridized. That looks like that transition state. What's going on at the action part of the mechanism up there? So it's going to be this one.
But how do we know if it's like non-competitive?
How do we know what kind of inhibitor it is? Good question. So we are binding, we're looking for the best inhibitor, right? And we're doing that based on the shape of what's binding the transition state. Where does the transition state get bound? In the active site. So we know that our inhibitor must be bound in the active site as well. Okay?
Can that make it mixed or competitive?
Sure. Was mixed one of our choices? I would still go with competitive. Okay. Yeah, I mean, it's in the, it's binding in the active site, right? Yes, anything could be mixed. I'm not going to give you credit for it. Okay, unless I give you a graph and it's mixed. And remember, after you've left this class and you've forgotten most of what we've talked about, and somebody asks you what kind of inhibitor something is, and you don't remember anything, you say competitive. Okay? It's most likely going to be correct. If you don't have any better hope. Other questions? Does that cover it? Okay. We still have 10 minutes left. Yes?
It's a quick question.
Hmm?
For the lipid-soluble vitamins, do you want
Yes.
us to memorize, I think it's 4 of them, or do you want us to just memorize one like we did with the...
So on the study guides, it says memorize 4 and tell a story. No. What does it say on the study guides? What was that?
You told us to know
like one?
Like a couple of that, like different lipids. You told us to know one of the steroid hormones, but you didn't specify for the lipid-soluble vitamins.
What does it say on the study guides? Does it say nothing? Okay, then 2. Somebody look that up and correct it if they want. But if whatever it says on the study guides is what I'll go by. Yes. Let's go to Sophia. I guess you had.
We talked about that one. Was it vitamin A that helps with your sightseeing?
Yes.
But then you talked about that one story in history,
Yes. You don't need a history story. Is that what you're asking?
Well, no, yeah, I was just curious. I looked at it and I forgot what
the story was. Let me take, I can tell you afterwards, or I can tell you guys if you're just looking for a story because you don't have any questions. But let's take Bridget's question then.
Can you go over I know the difference between the quickest amine glycans and the proteoglycan, but what do you just want us to know the difference or do you want us to know the whole heparin activating antithrombin thing?
I don't expect you to know all the details of that, but let's go over that because that'd be a good thing for us to go over. So we've got 2 things that are very similar and often people are confused on. We have proteoglycans. Okay, and what proteoglycans is, they are a combination of a protein and a carbohydrate. Okay, but it's a protein, and the protein's just an anchor. It's going to help hold this thing in the cell membrane. Protein is structural and acts as an anchor. All right, and I'll jump to a picture in a minute. Okay, and then we have the carbohydrate, and this is I'm going to say it's a carbohydrate, or I'm going to say it's a polysaccharide. but I'm really it's really something more specific than that. It is it's a glycosaminoglycan. Okay, and a glycosaminoglycan it's a repeating disaccharide that's highly negatively charged. All right, and so this does get to something. I think I'm going to correct what I said to you, Bridget, in terms of because we didn't clearly define what I said that you didn't need to know. Okay, I don't feel like you need to know very specific. You don't specifically have to tell me what heparin does, but you do have to be able to give me examples of how these things are functional, right? So this has the function. Okay, and I'll talk about what that function is in a minute. I want to define the other class here first. Then we also have glycoproteins. Okay, and the protein, they have proteins in them. Protein has function of some sort. Could be an antibody, could be these things, could be anything really. Okay, um, it also has an oligosaccharide on it. The oligosaccharides are highly branched, short, well, I'll say relatively short, sometimes really short, polymers. And they act as information-rich tags. Okay?
The difference between the two is that the polysaccharide has like the function in the proteoglycans, and then in the glycoproteins, the protein has the function.
Yes, but then the roles of so we could talk about the functions, but then essentially we can talk about that in more detail. So this is structural. We don't say much about the proteins here because the proteins can be all sorts of things necessarily. Now, this protein can certainly be all sorts of different types of proteins. It can be enzymes, can be things binding to things. Here, the proteins really anchor, okay, just structural. But these two, our carbohydrates here have different functions, and I care that you kind of know what they are. Okay, so here, this one's reasonably easy. It's a highly branched, relatively short polymer, and it's an information-rich tag. It's for cells talking to other cells, okay, or it's for well, it could be cells talking to other cells, but if it was in a membrane, if it's not in a membrane, it's a cell being able to recognize. So one example I gave you is the liver cell being able to recognize how long has that protein been in the liver. Or not been in the blood, not been in the liver, right? So how old is it? If it's really old, pull it out. Another example would be, you could put an oligosaccharide on a protein in a cell, and that would say, hey, you need to export this. It's a tag on the protein that tells that cell this needs to be spit out into the blood. Okay. But for the proteoglycans, we've got these highly negatively charged polymers. Okay. And what they do, think about the next lecture. No, really? Was this studying something? No. Did this other lecture end up in the wrong spot? It can't be that long. I guess we did those last, didn't we? All right. But I've got that picture that shows, I want to show you the picture where we got it's like this little hairy fibers on the outside of a cell. Was that earlier? Well, I'm not going to get it. Let's go ahead and let's talk about this. Let's make sure we get this in. So we have on the out. So heparin was an example of this, but essentially what we have is, I'm sure it's in the slides here somewhere, but I've got a protein, okay, and then I'm going to have glycosaminoglycans off of it, okay. In this case, it was heparin sulfate. Okay. On the outside of the cell, so this is our cell, and then this would be our cell surface. And so these collect things. So one of the examples I had, and you guys can go back and figure out maybe the next day, or somehow it's out of order here in terms of the notes. But one of the examples here was, antithrombin gets collected. It's positively charged, it gets attached to the negative charge, it changes shape, and then it kind of sequesters active factor X, which is AA, on its surface. Okay? You can also sequester, in terms of the blood clotting cascade, you can sequester thrombin. Lots of technical problems here. I can't lift it up. Can you guys see that in the back? Maybe you could stand up. You can sequester thrombin as well. Okay. You can also sequester. Our other example was lipoprotein lipase. So you want, if you want to break down triglycerols in the blood so that a cell can pick up those fatty acids, you have an enzyme called lipoprotein lipase. Let's just call it lipase. You have a lipase protein that does that, and it gets sequestered. You have these negatively charged, it is positive charged, and it gets sequestered here. And so what that helps make sure is that I don't have to make lipase so that it's at this super high concentration through my body. I just have to make enough that it can be sequestered on the surface of my cells, rather than have a bunch of it out in the middle here and have to have a higher concentration of it. I can have lipase on the surface of my cells so that when an LDL molecule goes by, okay, LDL complex might be a better way of referring to it, goes by, that these guys can grab fats out of them. Yes?
So are both of those pictures showing sodium bicarbonate?
Both of these are, this picture right here, this is my blue line. Mm-hmm. This right here. So this is the heparin sulfate. So this is kind of where the function of the proteoglycan would be, yeah. But all we're seeing in the picture are the glycosaminoglycan pieces. Yeah. Yes?
What are the dashes in the circles again?
What are the circles over here? These are lipases. And there is an actual official figure. So this is a cell, this is a cell, this is a cell. And the point is that we have these proteins sticking up, and it's like these hairy filaments out there that are negatively charged, help collect things to cells.
Is that what sequester means?
What sequester? Oh, thank you.
Is that what that means?
Good question. When I say sequester kind of means trap. Okay. Trap or store. All right. If you have more questions, feel